Hello and welcome back. Today we are diving into the first part of this subsection of notes. This is all about neuropharmacology, which is like the effect of drugs on the brain, but it talks so deeply about neurotransmitters. I wanted to sort of have a separate smaller set of notes that just sort of goes through different types of neurotransmitters. That way, when you are learning about them in the next set of notes, you're not like, well, wait a minute, what does all of this actually mean? So ideally, this will give you the background information that you may be missing at this point to understand neuropharmacology in its entirety. So let's start with going through the neuron again. So this is a neuron. The neuron receives impulses from other neurons that originate from the brain. These impulses travel through the neuron and then get passed on to the next neuron. The direction of nerve impulse always goes from a dendrite, which are these like branched areas around the cell body, through the actual axon, and then end at the motor end plate, in which there is a synapse, which we'll get into again, but in that synapse, the message gets transferred to the next set of dendrites, and it goes through over and over again. Now, on the axon, there are things called Schwann cells. Ooh, Schwann cells, named after, of course, the scientist. When you discover something, you name it after yourself. Why wouldn't you? So these Schwann cells, these are like the blue things that you see on the axon. They are encased with what's called myelin sheath. So myelin sheath is surrounding the Schwann cells. They wrap around the axon, and it helps move the impulse at a faster pace. In between the Schwann cells, like the little gaps in between, they are called nodes of Ranvier. I'll give you three guesses on who invented that, or who discovered that. So they are the uncovered parts of the axon. So while the Schwann cells in this picture do look like they're touching, and it looks like they kind of go like almost consecutively throughout the axon, there are little tiny gaps in between each Schwann cell, and those little gaps are the nodes of Ranvier. And what's cool about this is when you get a nerve impulse on the dendrite and it starts to travel through the axon, it sort of hops from Schwann cell to Schwann cell, so it like goes a little bit faster because it doesn't have to travel through the whole axon. It like skips over parts. And so myelin sheaths are like super helpful for like moving things faster. If you've ever heard of multiple sclerosis, which is a disorder, it's actually the breakdown of these myelin sheaths. So it impacts how quickly you can send impulses through the body. So continuing on, let's review nerve impulse. There is the resting potential and the action potential. The resting potential is sort of like where your nerves are at when they're not doing anything. So when a neuron is not firing an impulse, it rests at negative 70 megavolts, inside the cell is more negative than outside, and it's maintained by a sodium-potassium pump. This is just like a number you need to know, negative 70, that's just like resting potential. It needs to rise above a number, which I'll show you in a minute, to hit action potential. So this is when the actual impulse will go into the neuron, and the electrical potential across the membrane is reversed and then restored. So you are changing the charge of the neuron, and you are depolarizing it, and then once the action potential has passed through the neuron, you are repolarizing it. So depolarization is changed from a negative to a positive, and repolarization is changed from a positive to a negative. Okay? So you're starting at a negative, right? You start at negative 70, so it goes from a negative 70 to some positive number, and when you're depolarizing, and then when you're repolarizing, whatever that positive number that you were at, it will go back to negative 70. So here's just like an example of that. So like up here, negative 70 is your resting, and then you have to have enough of an electrical change in the impulse to go above a threshold, which in this case would be like 0, to change it to the positive number, and then depolarization will happen. So we've made it through the dendrites, we've hopped over the myelin sheaths, now we are at the end plate where synapses are. So synapses are the gaps between the end plate of one neuron and the dendrite of the next neuron. And so when an action potential reaches the terminal end of the presynaptic neuron, ooh, that's a lot of words. What does that mean? The action potential made it to the end of the first neuron, and now it's about to hop over to the second neuron. The neurotransmitters, the chemical messengers, they will diffuse across the opening here. So like this is the end of the first neuron. The chemical messengers will like get packaged up into little vesicles, and they'll go to the end of the first neuron. They will be released across the synaptic gap. Once they're released across the synaptic gap, they will bind with the receptors on the second neuron. And this area where they bind is the dendrite of the second neuron. So they go from the end plate through the gap to the dendrite of the second neuron. And they will continue on, they'll pass through, they'll go over the myelin sheaths of the second neuron and continue on. Now neurotransmitters can either be excitatory or inhibitory. Kind of makes sense, right? Excitatory contribute to the depolarization. So that means like, yes, 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 we love this, we're getting to the positive number, and they will continue the action potential, whereas inhibitory does the opposite, and it inhibits the depolarization of the neuron, and instead it will actually hyperpolarize it and go even more negative. And that will prevent the action potential from actually like firing off. So, I keep using this word. What are we talking about? What is action potential? So, we are going to talk about the action potential in steps right here. So, resting membrane potential, this is like your basic negative 70. You have more sodium outside than inside. So the outside is positive and the inside is negative. This is like when we're talking about all around an axon. Depolarization happens, so the membrane allows sodium to go inside of the membrane. Okay, we are getting more and more positive here, where the membrane is getting more positive. The inside becomes more positive and the outside more negative, right? Because those positive sodiums from the outside are now inside, so that positiveness is now inside. And the action potential travels as the sodium ions diffuse down the axon. So we started, I'll just go back one, right? We're starting here, right? We see that we're not really doing anything, but now we're starting to diffuse those sodium ions in. We're seeing that change as it continues to travel, right? So like now we're in the next step. Look at how this action potential is traveling down, because the gates in the sodium-potassium pump to let sodium ions in are opening. And as they're opening, the action potential is moving and moving and moving, and it's going all the way down the axon. As the change in positive and negativeness happens and the action potential moves down the axon, once it passes certain areas, repolarization starts to happen. So potassium ions rush out of the neuron, which makes the inside negative again, which is bringing us back down to our resting potential. After a while, well, not like, it takes like seconds, but in the world of like cell function, after a little bit, the sodium potassium pump will restore the original configuration, bring us back to homeostasis, and get prepared for our next action potential to come through. Okay? Now, this swap of sodium potassium happens at the nodes of Ranvier. Remember, nodes of Ranvier are those gaps in between the myelin sheaths, so this is like direct access to the axon. We don't have to go through the myelin sheath and then the Schwann cell and eventually get to the axon. If this only happens at the nodes of Ranvier, then transmission happens way, way faster because it's just one membrane that you have to work through instead of like 4. Okay, this is just sort of like another image, but we have our resting potential. Then we will have that depolarization start to happen where our sodium diffuses in. We make the inside more positive. We're getting positive, positive, positive. And what do you know? We have an action potential. Woohoo! Look at it. We're in the positives up here. Okay? As that happens and this action potential passes through the neuron, it needs to come back to resting. And so it will very quickly repolarize, and I do mean it will very quickly. Like, look at this is in milliseconds. Okay? So we're talking like two milliseconds, all this happens. So it will drop, 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 drop. Unfortunately, it overcorrects. So that's just something that will always happen. It will go too low, and this is when the sodium potassium pump will spend just a little bit of time getting back into our like homeostasis mode and going back into resting potential. Okay. This is a little bit more of like an image of synaptic transmission. This is how that action potential has traveled through the axon now. Now we're at the end plate of our first neuron. So what actually happens? Well, our axon terminal gets the action potential. So first the action potential has to actually reach this. Our second step is that calcium will start to come in to the end plate. So calcium starts to diffuse in. When calcium diffuses in, it's like a chemical signature that says, hey, start doing stuff in here because I'm here and like I'm your boss and it's time to start working. And once calcium tells everyone to start working, the vesicles will start to release those neurotransmitters. So the vesicles that are formed will go to the ends of the motor end plate, or the ends of the neuron, reach the membrane at the end, and release those neurotransmitters, which are these like orange dots. Those neurotransmitters will cross the synapse. They will bind to the neuroreceptors on the second neuron. So each of these green things is a separate neuron. So this is like the dendrite of a second neuron. And then once those neurotransmitters hit that receptor, it will trigger an action potential in the second neuron to continue that, whatever that impulse, continue it down all the way to wherever it's going. Usually, this could be anything really, but a very obvious one could be like sending a message like to your fingers to pick something up, right? You have to send an impulse from your brain. Your hand doesn't work by itself. It works because something tells it to work, and it's your brain. All right, so neurotransmitters again. There's excitatory, there's inhibitory. We're going to talk quite a bit about these different types of neurotransmitters in our next set of notes, but Basically, excitatory increases the permeability of the membrane, so it generates an action potential, it has the influx of sodium, and it causes depolarization, whereas inhibitory makes all the positive ions move out of the cell, depresses the postsynaptic cell. It's really hard to excite when you're dealing with inhibitory neurotransmitters. It takes a lot of neurotransmitters to get the excitatory reaction, the depolarization to happen. Really difficult. This is talking about chlorine in or potassium out. It just makes everything negative. And hyperpolarization happens where it goes even more negative. Okay, so decision-making in the central nervous system, when we're talking about how we actually send out action potentials, so one axon can feed multiple other dendrites. So when we're talking about this, right, we have how many is this? 5 neurons coming in, and so all of these axons are your first neuron, and they can feed into the dendrites of one singular neuron. Okay, so if you look at this picture, you have 4 out of 5 of them are positive and 1 is negative. That means that 4 out of 5 of these neurons are sending excitatory neurotransmitters and 1 is sending an inhibitory neurotransmitter. However, it doesn't really matter because the decision to send the next action potential is achieved by summation. So you add all of these neurotransmitters that were sent, and if it all adds up to above the threshold, then you will propagate an action potential. If it is below, then you won't. It just depends on what is actually being sent to the postsynaptic neuron or the second neuron. Where summation happens is called an axon hillock. It's sort of right before the action potential would go through the myelin sheaths. It has to actually pass and rise above the threshold. So there is a spot on the postsynaptic neuron that decides, am I going to continue this action potential, or am I going to just say we're done, we're inhibitory now, we didn't make it over the threshold, so we're gonna not pass this message along. So, types of summation. There's temporal and there's spatial. So temporal is rapid succession of action potentials arriving at one spot. So this would be one neuron attached to a second neuron, and it's just continuously sending the same action potential over and over again. And you can see here that it's a small jump, another small jump. By the third small jump, we've passed the threshold, so the second neuron is going to send its action potential. Spatial is sort of what we had talked about on the previous slide. So you get all of these action potentials from multiple sources. Some are excitatory, some are inhibitory. It just depends on what they all add up to see if we are going to actually continue on in this action potential. So continuing with this idea of decision making, if the summative effect reaches the threshold, the action potential will continue in the second neuron. And so in this case, you can see at the beginning, we have an excitatory, so ooh, a bump, yay, we're getting close to the threshold. And then we have an inhibitory, so we're going back down, even below resting. And then if you get an excitatory and an inhibitory, they usually cancel each other out. So we haven't hit the threshold. So in this first part, we are not, we're not actually hitting the threshold, so we're not going to send the action potential down to the second neuron. Whereas in this I don't know, blue, purple color, I don't know, you have excitatory after excitatory after excitatory, and it bumps, 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 boom. We got that beautiful action potential. It will continue on into the next neuron. Now, neurotransmitters can also be differentiated slow versus fast. So fast-acting neurotransmitters just have an effect within 1 millisecond of binding to a receptor. So they happen quickly. Slow-acting can take up to a minute. So it does have an effect. It's just slower. An internal second messenger molecule is needed to open the ion gate. So remember, the ion gate is your sodium-potassium pump. This is moving the sodium-potassium pump, continuing down the axon. So if it's a fast-acting neurotransmitter, it will just hit the target cell, and boom, we're moving, we're going down, we're opening gates, we're pushing that action potential, that impulse all the way down quickly. Slow-acting is like, hey, I showed up. How are you? Let's chat for a minute. I actually have a friend with me. Is that okay? And then the friend goes and opens the gate. And it just takes a while because, you know, you have to do introductions. It's like a whole thing, right? So slow acting, it's still, it doesn't take like days. It's like a minute is the longest it would take. And that's not that long, but it does take like hundreds of milliseconds usually, versus fast acting is like 1 millisecond. So slow acting neurotransmitters can modulate fast acting neurotransmitters. So when slow acting neurotransmitters are released into the cerebrospinal fluid, they can modify fast acting neurotransmitters. And they can do this by either regulating the efficiency of the release of neurotransmitters from the presynaptic neuron or regulate the efficiency of the postsynaptic neuron. So they can mess with the beginning, they can mess with the end, but it's all about regulating the efficiency. And so they can actually slow down fast-acting neurotransmitters just because they might make the neurons less efficient. Okay, so the last thing we're going to talk about is neurotransmitters affecting memory and learning. So this is an example that IB has that you need to know. It's about kind of seeing what happens to marine snails when we give them serotonin. Now, you've heard of serotonin. It's usually compared to like you feel good. When serotonin is released, it feels good in your brain, you're happy. It's like dopamine. It's all like, yay, we like to have serotonin in our brains because it makes us smile. So serotonin is a slow neurotransmitter, and so what scientists did was they gave snails a puff of serotonin. They basically just like exposed them to serotonin, and they saw that they had an increase of calcium into the presynaptic neuron. Remember that calcium is needed to send the vesicles of neurotransmitters to the next neuron, right? It sends it to the membrane. So when we see this increase of calcium, it causes that chain of events to occur, and it ultimately increases the amount of neurotransmitters that are released from the presynaptic neuron. So if the snail is stimulated once, right, it gets one puff of serotonin, the gill is retracted. That was like what they were studying. A second slight stimulation causes the same really strong reaction to occur. So even though we gave one puff of serotonin, we see this gill being retracted, great, that's what we wanted to see. We can do a much smaller second stimulation and we'll still get the like extreme reaction of it. And so what we're seeing as scientists, what the result is that we just saw short-term learning. We just saw a reflex happen, right? The gill withdrawal reflex. The snail had learned that serotonin equals gill retraction. Okay? So it's a short-term solution, but it is something. So then they were doing more and more experiments with this snail. And so they wanted to see, well, that was short-term memory. Let's look at long-term memory. So to create long-term memory, you needed a longer lasting stimulus. In this case, instead of 1 puff of serotonin, it was 5. When the snails were exposed to 5 puffs of serotonin, the genes in the nucleus were activated. So this serotonin caused actual protein synthesis to occur. And these proteins changed the shape and function of the synapse that was there to retract the gill. And by changing the shape and function of the synapse, it made additional connections with other neurons to allow this to happen at a faster rate. So this is just sort of like showing you that as you continue to do something over and over again, or increase, in this case, like the amount of serotonin that is causing some sort of reaction, you will eventually actually reshape your postsynaptic neuron or make additional connections. So in this case, you can see that the shape was modified. And this is not something that happens like all the time, but it happens fairly frequently, especially when you're making long-term memories, like how to ride a bike, or how to write in cursive, or something like that. Even things where, like for example, when you were a kid, maybe you learned how to play piano, but you haven't played piano in a decade. If you sat at a piano, you could probably pick out middle C still, even though you don't know maybe how to play piano anymore. You might still be able to read like an easy sheet of music. And that's because those connections still exist, and they probably will easily be refound because there were so many connections at one point to make this long-term memory. Okay, the last thing we're going to talk about is the reflex arc, which is how your body actually interprets a stimulus, whatever that stimulus may be. In this case, we are talking about a pin. So this is like, oh no, I stabbed my finger with a pin and it hurts. So when that pin actually goes through and into your skin, there are receptors in your skin that feel that and say, whoa, this isn't normal, and I'm going to send a message saying there's something here, and we don't like it. And so that message gets picked up by the receptors, sent through a sensory neuron. That sensory neuron will go all the way to the spinal cord. There's multiple neurons here. This is not like one singular neuron. It will continuously get passed. And when you get to the spinal cord, there is a relay neuron. That relay neuron might send a message up to the brain to be like, hey, what do I do? Or it might just be like, whoa, this is really bad. I'm going to immediately send a response down to the motor neuron. And so, again, sensory neuron sends the information. Relay neuron decides either to send it to the brain or to continue on and skip the brain because it's like really bad. And so the relay neuron will pass on the message to the motor neuron, which is this blue one. And again, it might have multiple neurons. This is just like a picture. And the motor neuron will connect to an effector. It's a muscle, but it's like the end result. And so once the motor neuron gets the message from the spine, it'll remove the stimulus if it's painful, or like if you put your hand on a hot stove, it'll lift the hand off of the stove because like, ow, that should not be happening. And so this is how you get a response. So there's a stimulus, gets picked up, sensory neuron to relay neuron to motor neuron to effector at the end. Okay, that's everything for this one. I know it was a little bit long, but it will make next set of notes much easier. I hope this made sense. I will answer any questions in class. I'll see you then.